Welcome to today's video tutorial in which we'll be studying about dark field and phase contrast microscope. Okay, let's get started. The first type of microscope is the dark field microscope. Okay, so as the name tells here, dark field means, so when you observe the specimen under this microscope, the background appears completely dark or completely black and the specimens will be illuminated over here. Okay, and that's the reason we call it as dark field microscope. And so when do we use this dark field microscope? So it is used for examining the live organism. So when you want to see the organism live, when you want to see them moving, okay, so then you can use dark field microscope. And secondly, it is mainly used to see the appendages of the organism. There will be appendages in the organisms, right? Like for example, flagella and all those things. So that will be seen very clearly in the dark field microscope. Okay, it will give a good contrast. And apart from that, I think there are many other organisms where if you stain, so it will be distorted and the characteristics cannot be visualized properly. So in such cases also we use this dark field microscope. Okay, so it is one type of microscope. And there are many other types of microscope like differential interface contrast microscope, phase contrast microscope, fluorescent microscope, electron microscope. All these things we'll study one by one. So, but among them, one is dark field microscope. Okay. So now moving on to the principle of dark field microscope. So this is a ray diagram of dark field microscope. So this is how it looks. So now this actually works similar to a bright field microscope. I think in the previous video we studied in detail about the parts, about the working principle of the bright field microscope, right? So this also works in the similar manner. So but it has an extra component which is called as the opaque disc. Okay, so now rest all are the same. So here there will be light source, there is condenser, there is specimen, there is objective. So everything is similar to bright field microscope, but the additional extra component is this central opaque disc. Okay, so this changes the principle of the microscope. Okay, so now as you can see here, so this is the disc which is circular in shape. Okay, and so there is blocked out region in between this disc and also there is blocked out region around the disc. So you can see the black portion over here, no? So this is the blocked out region and the outer thing is also the blocked out region. The blocked out region means over here the light cannot pass through. Now for example there is light source over here. So the light is getting produced. So but the light cannot pass through this blocked out region from here and from the outer edge. So whereas so there is one small portion over here which is unblocked in between. As you can see here, in between there is a small unblocked region and the light can only pass through this unblocked region. So you can see the light is only coming out through this unblocked region. Okay, so now this light, a small amount of light which is passed through this unblocked region, no? So it reaches the condenser. So condenser is similar and it works similar to our bright field microscope. And so again from the condenser the light hits onto the specimen. The specimen is over here. Okay, and we all know that so the refractive index will vary based on the components. Okay, so now different components will have different refractive index. So the same thing over here. So when it hits the specimen, no, now the slide is completely made of glass and the specimens over there will be of different components like different proteins and many other components will be made up of. As a result there will be change in the refractive index in the background and change in the refractive index in the specimen. As a result what happens, the specimen will start producing or scattering light. Okay, and that scattered light will actually reach the objective. Okay, and the thing is it is designed in such a way that so the other light, that is, no, for example, light is from the unblocked region is reaching the condenser, and then it is reaching the specimen, and from this specimen it will be diffracted, that means it will move out. Okay, and this particular diffracted light will not reach the objective, whereas only the scattered light from the specimen will reach the objective, as you can see here. So there is a small amount of light passing through the objective, so this is the light scattered from the specimen, whereas the other part of the light which is diffracted will move out, so it will not reach the objective. And as a result, no, so only the light scattered from the specimen will reach the objective, which will reach your eyesight. So as a result, you can find the good contrast, so in which, so there will be a very dark background and the image will be illuminated beautifully. Okay. Okay, so now, yes, so these are some of the pictures or images of the dark field microscope. As you can see, the background is completely dark and you can beautifully see the specimen over here which is illuminated. See, you can also find the color, the natural color. It's not stained over here. So, but you can also find the natural color of the specimen in the dark field microscope, which is beautifully illuminated. Okay, so these are the images of dark field microscope. And yes, so uses of dark field microscope, as I told, so it is mainly used for studying the live organisms. Okay, so you can find the organisms moving over here, you can see the microbial motility over here, and also you can find the tufts of the bacterial flagella, that is, you can find the beautiful structure, very detailed structure of the flagella. So even if the flagella is in bunch or it is in tuft also, you can see them very clearly in the dark field microscope. And apart from that, we also use to visualize spirochetes, mainly like Treponema pallidum. Okay, so and then Borrelia Burgood Ferry and Leptospira interrogens. So all these, no, all these spirochetes can be very beautifully seen under dark field microscope, because when they are seen in the light field microscope, no, so they cannot be visualized very clearly because they are very thin, they are very slender. So and that's the reason we cannot observe in the bright field microscope so clearly. So whereas in dark field microscope they can be seen very clearly. So these are some of the uses of dark field microscope. Okay, and now coming to the advantages of dark field microscope. So yes, so compared to the bright field microscope, no, yes, dark field microscope's resolution is far better. Okay, and so the thing is, we do not stain the organisms over here. We need not kill the organisms here. You can see the live organisms here, and we do not have the procedure of staining for this. And even you can directly detect the unculturable bacteria present in the patient sample. Okay, and so here, yes, preparation step is not required here as much as bright field microscope or staining procedure. Okay, and yes, if you want, you can use by bright field microscope, and bright field microscope itself can be changed to dark field microscope just by the addition of the opaque disc. Okay, that is another advantage over here. So just by placing that opaque disc in the bright field microscope, it becomes dark field microscope. So all these are the advantages of dark field microscope. Now coming to the limitations of dark field microscope. Yes, here sometimes what happens is, when the sample, no, it is illuminated very strongly, that is when the light scattering is done a lot, when it is illuminated strongly, no, there is a chances that sample might get distorted or sample might get damaged. Okay, and apart from this, so sometimes what happens is the dust particles, so accidentally there is some dust particles in this slide, so the particles also will start illuminating, they also will start scattering the light, and so they also start appearing bright. So that time you cannot differentiate whether it is organism, whether it is dust particle. Okay, so that is one limitation or disadvantage over here. And then the thing is, you have to take a very thin film when you want to observe in the dark field microscope. Okay, so if it is even a little dense preparation, no, so you cannot find that contrast, or you cannot find the effect of the contrast and the accuracy also. Okay, and as a result, very thin spread has to be there. Okay. And yes, it has to be done very quickly also. Since it is a live organism, you have to do it quickly. So all these are the limitations of dark field microscopy. Okay, so next type of microscope is phase contrast microscope. So yes, this is another type of advanced microscope, so which helps you to obtain a very clear picture of living or unstained cells. Okay, so yes, so when you take bright field microscope, there you need to stain the specimen and then you need to visualize them under microscope. Okay, so when you try to visualize unstained living cells, know what happens? This unstained cells, they don't absorb any light. Okay, and because of this poor light absorption, no, so it results in very small differences in the intensity distribution. Fine. So and because of this, no, so you can barely see the specimen, or sometimes you cannot see the specimen at all. So hence it is compulsory to visualize the stained cells in bright field microscope. So to overcome this, yes, we have microscopes like dark field microscope, phase contrast microscope. So compared to dark field microscope, yes, phase contrast microscopes gives you a much good contrast, and it gives you a much clearer picture. Okay, and here mainly phase contrast microscope, no, it works on the technique that converts phase shifts into the light passing through a transparent specimen to brightness changes. Yes, so mainly here it works on the principle of phase shifts. Okay, or I can tell brightness changes in the slide. Okay, and this particular phase contrast microscope was described in 1934 by a Dutch physicist Fritz Zernike. Okay, and so here in we studied that in dark field microscope we use just a single extra component which was called as annular ring. Okay, so whereas in this phase contrast microscope we use a special condenser and objective lens with a face plate to convert these phase differences into visible differences in the brightness or amplitude. That is basically, if I want to tell, so we use two components over here, two extra components. One is the annular ring, so just as in dark field microscope. Apart from that, we have face plate in the objective lenses. In the objective we have face plate, so this two components is the one which creates the phase shifts. Okay, and so provides you the image. So we'll study about them in detail. Okay, so now coming to the principle of the phase contrast microscope. So this is a ray diagram of it. So yes, so coming to it, so first is the lamp, so which produces light. So and then comes the condenser annulus. So yes, this is the extra component which we can see in the phase contrast microscope. And as you can see, so this is the one which is allowing the hollow cone of light to pass. Okay, and here, so just like in dark field microscope, we'll have a blocked out region around and blocked out region in between. Okay, and only there will be a small portion of unblocked region in between over here, and through this, from this the light actually passes through the condenser, and from the condenser the light hits to the specimen. Okay, so once the light hits to the specimen here, so you can actually um, expect two types of light over here. Okay, so that is undeviated light and diffracted light. So what is this undeviated light? So undeviated light, so is also called as zero or zeroth order light, or you can tell it is a surrounding light. Okay, and so why do we call it as undeviated is because this light passes straight through the specimen without being scattered or altered. Okay, or I can tell it essentially acts as a background light in the final image. It is passing through the specimen, or it is mostly it is passing through the slide, and it is not getting deviated, and it acts as a background light here, so that is called as undeviated light. And so there is another type of light which is called as diffracted light. As the name tells here, diffracted light are, see, as you can see here, so diffracted light is the light that has been scattered by the specimen. Okay, light is typically passing through the specimen, so it is getting scattered by the specimen due to the interactions with its internal structures such as organelles or other components and all those things. Okay, and here I can tell this undeviated light mostly passes through the slide, and that's why it will not get deviated, and hence we call it as undeviated light. Whereas this diffracted light, no, so yes, this diffracted light is the one which passes through the specimen, which interferes with the organelles and all those things, and as a result it is getting scattered. Okay, so hence, so once it moves through the specimen, so you can find both diffracted light and undeviated light. Okay, so and yes, this diffracted light, no, so it spreads out in various direction as it passes through the specimen. Okay, and the thing is, now both these undeviated light and diffracted light, no, so they have a way, that is they take a separate time to pass through. Okay, that is I can tell that the light waves passing through a specimen, okay, so can be slightly delayed or it can be advanced in phase when you compare to the light through the surrounding medium. Okay, so that means I can tell uh, in a more simpler way that now the light passing through the specimen, no, so they will come out late. The light will slowly pass and it will take time to come out. Okay, so whereas when you take the light passing through the surrounding medium, so they come out very easily, they come out fast. Okay, so as a result I can tell that undeviated light will pass faster when compared to the diffracted light. Okay, so this actually creates a phase shift. Okay, so when both these lights passes through the special objective, okay, that is we have a special component in the objective called as phase plate, no, so when it passes in the different time, so they create a phase shift and they can be observed easily by using this phase plate. Okay, so this phase plate is also similar to this annulus, and so it can make you clearly see the contrast. Okay, because of this phase shift you can easily see the contrast. So this is the principle of phase contrast microscopy. Okay, and then, okay, as you can see here, so this is a picture of a face plate in the objective lens. Okay, and yes, next is the application of phase contrast microscope. Yes, we to produce the high contrast images of transparent specimens such as living cells, microorganisms, thin tissue slices, lithographic patterns, fibers, latex depressions, glass fragments, and subcellular particles including nuclei and other organelles also can be visualized by using phase contrast microscope. Okay. Okay, and then coming to the advantages of phase contrast microscopy is, so like bright field microscopy, you need not stain here. Okay, and staining, fixation, and all will be time saving over here, and so it makes a highly transparent object also visible. Okay, and so you can also see intracellular components like mitochondria, mitotic chromosomes, vacuoles, and all those things very clearly with a high resolution over here. So as you can see, this is one picture of the image which is obtained in phase contrast microscopy. Okay, and so you can also know how they that is living cells, how they proliferate through the cell division. Okay, and apart from that, yes, so bright field microscopy also can be converted into phase contrast microscopy by adding the components. As I told, there are two extra components. By adding that, you can convert the bright field into a phase contrast microscope and you can observe them very clearly. Okay, and then, Okay. Again, you can visualize two other images, so which is obtained in phase contrast microscopy. So yes, you can see the clarity over here. Okay, so how nicely you can differentiate between the organelles, between the surrounding area, cells and all those things. So clearly you can visualize over here, right? So this is better when compared to bright field and dark field microscopy. Okay, so but yes, it also has certain limitations like so the objective lenses and the condensers which we use and the cost of this no so is high. Okay, so because of the cost, yes, we don't usually use it in any other area. So it is just used in health professions by health professions, but we don't often use it in the teaching labs. Okay, because of the cost of the. And the second thing is here the to use phase contrast. Now the light path must be aligned. So yes, because as you told, there is blocked region, unblocked region, and all those things. Hence, the alignment of light is very much essential over here. And more than everything, more light is needed in the phase contrast microscope. Okay, than bright field microscope. So these are the limitations of phase contrast microscopy.